several traits. Like you can see that in pea plants we can have different colors. Okay, different colors of seeds, like yellow seed, green seeds, different colors in flowers. Okay, different colors of pods, tall plant, short plant, shape of seed. So different traits can be observed. Okay, in several traits. If I talk about that is several traits are observed or are observed observed in pea plants. Okay, are you getting my point? So what is the benefit of this several trait? The benefit of this several trait is that you can actually means a study means it will going to help our in studies. For example, if you are going to study only one character, for example, if there is a plant that don't bear different kinds of seed, that the shape is almost same of all, so you will not be able to understand that which character is being transferred, which is not being transferred. Are you getting my point? Are you getting my point, Saima, Shweb, Tejasvi?
Yes,
Okay,
sir.
so we will say that different characters were not observed there. Okay, if there is a plant, but in pea plant there are so many characters are there, and so many characters are going to help us in understanding better way. Okay, second is pea plant has bisexual flower. Don't write right now, I'm going to help you. Okay, you're going to write a different statement. So what is the benefit of bisexual flower? Bisexual flower, bisexual flowers enables us to self pollinate, means if we want to do the self pollination, so self pollination is possible. Self pollination is possible. The second thing is that we can do the cross pollination also, because it bears large stamens with bisexual flower with large stamens, stamens mean stamen is the male reproductive part. Okay, that that can be easily cut and removed. Okay, so if it is going to bear large stamens, so what is it, what is the useful things for having a large, or what is the benefit of having large stamen? That they can be easily cut and removed, can be easily cut and removed. Okay, and if they are removed, then what you can do, you can do the cross pollinations. Okay, so this make us cross pollination is possible. What is cross pollination? That transformations of pollen from different plants, or transformation of pollen grain from anther to stigma of different plant. So if we want, this is a plant A. Okay, plant A, and this is plant B, and if I want to do the cross pollination, I need to transfer the pollen grain from A to B. So what I need to do is we need to remove the anther. We need to remove the stamens. Stamens, they bear large stamens. Large stamens means male reproductive organs can be easily cut, and we can say that it will help us in tell me in cross pollination. Okay, got it. Third thing is that pea plants have short lifespan. Short lifespan. What is the benefit of short lifespan? That short lifespan is going to help pea plant. So we can say that it going to help the Mendel for studying, or in studying different or several generations in a short span of time. Okay, so if short span enables the observer that he can study several generation in short span of time. Okay, so several generation can be studied. Several generation can be studied. Okay, due to short lifespan, several generation can be studied. Okay, are you getting my point? Tell me fast.
Yes, sir.
Tejasvi, Shweb, Shweta, are you getting? Okay.
Yes.
Next is the pea plant bears large number of seeds. Pea plant bear large number of seeds. Okay, and what is the benefits of large number of seeds? That authentic conclusions can be understood. Okay, when we have, for example, if like mango tree is going to bear only one seeds. Okay, the fruit is going to bear only one seeds. So you cannot have large number of observation in that one. Okay, but since pea plant is going to bear large number of seeds, so we can say that out of 400 seeds, okay, out of 400, this much is round, this much is green, this one is yellow, this one is wrinkled, this one is round. Okay, so means authentic conclusions can be observed. So large number of seeds help help in getting authentic conclusion. Authentic conclusions. Okay, are you getting my point? Tell me fast. Are you getting my point? Are you getting my point?
Yes, sir.
So why do Mendel choose pea plant for his experiment? Do you know the why do he choose pea plant for his experiment? Tell me fast. So first of all, pea plants have short lifespan. The very important thing is that it has short lifespan. What is the benefit of short lifespan? That several generation can be studied in short span of time. Are you getting my point?
Yes, sir.
Are you getting my point? That several generation can be studied in short span of time. It has large number of traits. So what is the benefit of getting large number of traits? That it will help us in studying more traits. Okay. It helps us in studying more traits and more details, that how the characters are being transferred from one generation to another generation. Are you getting my point? Tell me fast.
Yes, sir.
Okay. So this was, and the next is, so please note it down that why do Mendel choose pea plant for his experiment? So wait a minute. Having the notes. I have shared my notes on your uh, group. Okay. So please note it down this one. This is okay. Wait a minute. No, please note it down. Why do Mendel choose pea plant for his experiment? So please note it down. Mendel choose pea plant for his experiment because availability of detachable contrasting characters, several characters like height, shapes, seeds, colors. Okay. Short lifespan, therefore several generation can be studied. Third is normal allows self-pollination because but cross-pollination can also be carried out. Why normal it allows self-pollination? Because it is bisexual. Okay, but cross pollination can be carried out because it bears large stamens. So please note it down. Why do Mendel choose pea plant for his experiment? Note it down. Done. Noted.
No,
Note it
sir.
down. Note it fast. When you're done, please let me know, okay?
Done, sir.
Other students,
No, sir.
note it fast. Done? Okay. Done, everybody?
Yes,
Is there
sir.
anyone who's writing? Done? Okay. Why are you not responding? Everybody, please turn on your camera. Turn on your camera. Tejasvi, Shweb, Shweta, Saima, turn on your camera, please. Next. Next is, if I'm talking about this one is, just a minute. Okay. Next thing is that we are just going to study the monohybrid and dihybrid cross. Okay. So why do Mendel do this type of experiments in monohybrid and dihybrid cross? So we're going to discuss this one. Okay. So Mendel's monohybrid and dihybrid cross. So what is monohybrid cross and what is dihybrid cross? Mendel's monohybrid cross. Hybrid monohybrid cross. So if I talk about monohybrid cross, what do you mean by the Mendel's monohybrid cross? Okay. So if I talk about this one is mono means single, or we can say that monohybrid cross is the cross in which we are going to study only one type of characters. And each character has at least two traits. There can be so many traits in a character. For example, if I'm talking about that, like height is a character. Height is a character. So we study two traits in it. So this is a character. This is a character. And this one is height is, what is height here? Height is a character, and dwarf and tall are the two traits. Okay. So what is monohybrid cross? The cross in which one character or one pair of trait is studied. Okay, is studied. So we called it monohybrid cross. Like one pair of trait. This is one pair of traits or one character we are studying. We called it monohybrid cross. So what does Mendel do actually? In monohybrid cross, Mendel took two plants. One is tall plant and another one is dwarf plant. So he took a plant which is approximately 6 feet high. Okay, 6 feet high plant, pea plant. Okay, and he called it homozygous tall. And another one was 1 feet plant, and this is 1 feet, and this is 6 feet. Got it? So here is 1 feet, another one is 6 feet plant. Got it? And what he did here is he was doing cross pollination. He was doing cross pollination. In the first generation, To obtain the first generation, he did the cross pollination. So he did the cross pollination. Okay. And when he did the cross pollination, he get all the plants of 6 feet. Means every plant was of 6 feet. Every plant was of 6 feet. All plants were of 6 feet. Okay. All plants were of 6 feet. So every plants. Okay. So what are the results of cross pollination in F1 generation? So this we called it F1 generation. This is F1 generation. And in F1 generation, all plants were tall. So what conclusion can you predict from this one? That there is no dwarf plant. What do you mean by this one? That traits are not intermixed. Okay. Traits are not intermixed. There is no 3.5 feet plants. For example, 6 plus 1 divided by 2. We should get an average of 3.5. But there was no 3.5 feet plants. Means that the traits or the genes are not intermixed. Okay. They maintain their identity. They are not intermixed. Got it? Shweta, are you getting? Saima, Shweb, are you getting? That
Yes,
traits
sir.
are not intermixed. Got it? They maintain their identity. Okay. And always remember that whenever he is doing the cross pollination, we are getting, whether it is a monohybrid cross, whether it is a dihybrid cross, we are getting the same results. All were same. Okay. Like here, we all are tall plants. And he called it homozygous tall. This is homozygous tall. Homozygous tall. And this is homozygous dwarf. Okay, homo means same, and we are talking about that what is homozygous and what is heterozygous. We're going to discuss this one. But in F1 generation, he is going to get all plant as tall plants. Okay. Next is when he self-pollinated. When F1 generation is self-pollinated, like this is a plant. Okay. And when it is self-pollinated, means the flowers of this one, okay, is being means self-pollinated. So during the self-pollination, when this F1 generation is self-pollinated, then for every 4 plants, we were getting, this is the F2 generation, we were getting 3 tall plant, okay, and 1 dwarf plant. 1 dwarf plant. So out of every 4, we were getting 3 tall and 1 dwarf. Got it? This is F2 generation. F2 generation. So F2 generation is obtained. Tell me when is F2 generation obtained? F2 generation is obtained when F1 generation is self-pollinated. Okay. So here self-pollination is taking place. Okay. Self-pollinations. So we obtained the F2 generation in which out of 3 plants were tall, 3 were tall and 1 was dwarf. Got it? Got it? So how do Mendel explain this one in terms of traits? So he said that when he take the traits, for example, he took the in F1 generation, he took the homozygous tall and homozygous dwarf. So this is capital T is homozygous tall and small t for the homozygous dwarf. Okay, got it? Capital T he took for tall plants and small t he took for dwarf plants. Okay, because we are going to have the pairs now, genes are in pairs. So when these plants are going to bear the gametes, so their gamete will have either capital T or small t. Like in human beings, if we have 46 chromosomes, so 46 out of 46 chromosomes, 44 are autosomes and 1 X and 1 Y is there in males. So when sperm formation is taking place, then it is going to have 22 autosomes, either X or 22 autosomes, either Y. It cannot have both. Got it? So same thing is here, that capital T, when it is going to have the gametes. So this is capital T, another is capital T, and it is small t and small t. Okay. So when they are going to mix, they are going to make bond. Okay. Or they are going to fuse together, fertilize together. Then the generation or zygote will have capital T and small t. This will be going to have capital T and small t. This is capital T and small t. And this is also capital T and small t. So we are going to have all tall plants here. It is also having that gene of the dwarf plant, but since the tallness, he's called that in heterozygous condition. This is not the this is homozygous condition. This is homozygous tall. Homo means same. Zygous means zygotic condition. Homozygous tall, which is going to have the same. And this is homozygous dwarf. Homozygous dwarf. So what does Mendel conclude here? That, and this is the heterozygous. It is heterozygous tall. Heterozygous tall. Okay. Why? Because this capital T and there is capital T and small t there. Capital T and small t. So the gene of dwarfness is also here. And they are not same. That's why it is considered to be as hetero means different. Hetero means different. Are you getting my point? Shweb, samajh aa raha hai? Are you getting? Saima? Shweta?
Yes, sir.
So this is going to have hetero means different. Zygous means, so this is different. Capital T and small t means one tall gene of tallness and another gene is of dwarfness. This is heterozygous condition, but it is showing tall. Still, it is not showing average plant. They are not mixing together like this. They are, okay. So still, why it is showing tallness? Because here the gene of tallness is dominant over the dwarfness. They are remaining together in an individual. They are remaining together in a plant, but here the tallness gene is dominant. Means it is showing, it is expressing itself in front of in front of the gene of dwarfness. Okay. So instead, the plant is going to have both the genes of tall and dwarf, but the plant is still tall. It means the gene of tallness is dominant over the dwarfness. Got it? Got it? Here also, capital T, small t, gene of tallness is dominant over. So all plants in F1 generation, this is F1 generation, all plants in F1 generations were tall. Why? Why they were tall? Because there is a factor which is dominant, the factor of tallness, the gene of tallness. Mendel called gene in his term as factor. Now this factor is modern term known as gene. So the gene or the factor which is responsible for tallness is dominant over dwarfness. Are you getting my point? Are you getting my point? Tell me.
Yes, sir.
Okay. Now when he when he self-pollinate this, self-pollinate means capital T, small t and capital T, small t. So this is capital T, small t, capital T, small t. Okay. So when this is going to fuse with this one, we are going to have tall plants. We are going to have tall plants. Okay. This is capital T, then capital T, small t, then capital T, small t. Okay. And then small t, small t. So out of three, this will gives you tall. This will also gives you tall, and this will also gives you tall, but this will gives you dwarf. So these three were giving you tall because here the tall gene of tallness is dominant over the dwarfness, and this will gives you dwarfness. So if I talk about that, this is F2 generation, and the ratio of F2 generation, we called it phenotypic ratio. Phenotypic ratio. So phenotyping, physical appearance. So if I talk about phenotypic ratio, ratio of monohybrid cross in F2 generation. In F2 generation is what is that? That is 3:1. 3 tall, 1 dwarf. But if I talk about the genotypic ratio is actually this. You see here, this is tall, then this is different from these two. So these two are similar and these two are different. Okay, so the ratio is, the genotypic ratio is 1:2:1. Got it? Got it, everybody? Got it? Please make this one. Monohybrid cross. Have you made this one? Made this one? Fast. Tell me.
Done,
No,
sir.
sir.
Make it fast. Make it. Make it.
Done, sir.
We have different results in F2 generation. Okay. Not in the F1 generation. F1 generation is going to have the same results, but in F2 generation, we have different results. Okay. Got it? Three were tall and one was dwarf. Okay. Got it?
Yes, sir.
Done this one. Why are not the students are not responding? Saima, Shweta, Shweb. Why are you not responding?
Done, sir.
Make this one. Make this one fast. Done? When you're done, please let me know.
Done, sir.
Okay, done. Draw this one also. Done? Tell me fast.
Yes, sir.
Yes, sir.
Shweta, done? Saima, done?
Yes, sir.
Yes, sir.
Okay. So what is the conclusion of Mendel monohybrid cross? Monohybrid cross. So first let's have the question here. Uh, uh, Here is a cross between a tall pea plant and a short pea plant resulted in a progeny that were all plants were tall. Why? Why? What was the conclusion? Because actually we were having capital T and small t there. We were having capital T and small t there. Okay. Capital T and small t there. This is a heterozygous condition. And in heterozygous condition, we are going to have one tall gene and one dwarf gene. But still the plant is tall. This shows that tallness is dominant over the dwarfness. Okay. Got it? So there is a gene which is tall, which is of tall, and it is dominant over the gene of dwarfness. So what will you answer for this question? Which is the correct answer?
A.
A. Tallness is dominant trait. Shortness is dominant trait. Tallness is recessive trait. Height of the plant is, yes, it is. Tallness is dominant trait. That's why we were having in monohybrid cross in F1 generation, we're going to have all tall plants. Okay. Got it? Everybody understood?
Yes, sir.
Understood? Okay. So we are going to have, if a question comes in examination that what are the conclusions of monohybrid cross? So the conclusion of monohybrid cross is monohybrid cross conclusions. Conclusions. Okay. What were the conclusions of monohybrid cross? What were the conclusions? He gives the principle of dominance, law of principle of dominance, or law of dominance, or principle of dominance. We call it principle of dominance. Okay, and what does this principle of dominance says? That in a heterozygous condition, the two different traits remain together in individuals. Okay, in a heterozygous condition, the two different traits remain together in individual and they are not mixed up. Okay, there is no average plant gene, so they are not mixed up. Okay, and the trait which is able to express itself is called dominant trait, and the trait which is not is called recessive. Got it? Got it? So what he said that in heterozygous condition, the two different traits, these are the different traits. Okay, they remain together in an individual. But the one trait which is able to express. Now here the tallness gene is expressing itself. The plant, when this condition satisfied, then the plant is tall. So it means tallness is dominant over the darkness. So the gene which is able to express itself is called dominant trait or dominant gene. Dominant trait or gene, if you are talking about the gene which is able to express itself is called dominant gene. and that is not able to express itself is called recessive gene. Okay, are you getting my point? So he gave the principle of dominance. Okay, so let you write the principle of dominance. Just a minute. Please note it down.
Done, sir.
Done. Noted. Noted, everyone. Shweta, Shweb,
Done,
Saima.
sir.
Noted.
Yes, sir.
Okay. So let's move further. That he also give another law, that is we called it law of segregation. Law of segregation. Okay, or law of purity of gene. Law of purity of gene. And what does it states? It states that in a heterozygous condition, the same condition, in a heterozygous condition, the two different traits remain together in an individual. You can see that they are, okay, they remain together in an individual, and they are not mixed up. They are not mixed up like this. Okay, and how will we prove that they are not mixed up? Because when we are going to have when we are going to have a cross pollinations of homozygous tall and homozygous dwarf, okay, plant, then we were not getting any average plant. Okay, if there is an average plant, For example, 6 feet plant is crossing with the 1 feet plant. Okay, then we should have a 3.5 feet or less than, means average plant should be there, but all plants were approximately 6 feet. There was no average plant or intermediate plant, so it means that they are not mixed up. Okay, they are not mixed up, and they maintain their identity. How we know that they are maintaining their identity? They are not forgetting. So like here we know that capital T in a small t. Just look at this, just concentrate here. When heterozygous condition is there is a capital T and a small t, and in this condition we get a tall plant. Okay, tall plant. Okay, but when we are going to have condition of small t and small t, then we see a dwarf plant. When we see a dwarf plant, okay, so it does not means that plant forget how to dwarf. Okay, they are not maintaining they are not forgetting their identity. This gene is for dwarfness and it is still knows how to dwarf. But since here it is with capital T, the gene of tallness, and this gene is actually dominant gene over this one. So that's why it is not able to express. It is not able to express. This is not expressing here. But it does not mean that this gene has forgot. This does not mean that if it is this gene is living with capital T, so like we have a good friends, okay, and bad friends. So if a person, if there is a good person, he start living in a bad friend, then he forgot his identity. Okay, maybe. Okay, he start doing activities with those who is sitting with him. Okay, or he's making friend with them. But okay, but here is not true. But in this case, it is not true. If it is with tall, this gene is of darkness is with tall, the gene of tallness. So it does not mean that it has forgotten its identity. When this and this condition is satisfied, when small t and small t are going to mix together, then we are going to have a dwarf plant. Dwarf plant. So what does law of segregation says? That law of segregation says that in a heterozygous condition, the two different traits remain together in an individual and they are not mixed up. They are not mixed up. They are not mixing. We are not getting an average plant. Okay, like this. They are not mixing. They maintain their identity. How they maintain their identity? Whenever this is a tall, small t meet together, they form a dwarf plant. So it means that small t is not small t has not forgotten its identity of dwarf or dwarfing. Okay, this gene is responsible for dwarfness. So it will dwarf the plant if we have a small t. Okay, so it has not forgotten its identity. Okay, it has not forgotten its identity. Okay, whenever small t meet together, we will going to have. Okay, and during the time of gamete formation, like when we have capital T and small t, okay, so during the time of gamete formation, then each gamete is going to receive a particular gene. Like I was telling you, like we have in males, we have 46 chromosomes. Out of 46, we have 44 autosomes and 1X and 1Y in males we are talking. So when the gamete is being formed, and during the time of gamete formation, we have 22 autosome, and either the sperm is either going to have X means it is 23, or it will have Y. It cannot have both. So similarly, during the time of gamete formation, when the heterozygous condition is there, capital T and small t is there. So when the gamete is formed, so either it will have capital T or either it will have a small t. Okay, got it? Got it?
Yes,
Got
sir.
it? So this is the results of monohybrid cross that we were given a law of segregation, law of purity of gene, that genes always remain pure. They are not mixed up. They didn't forget their identity. When the gamete is formed, then each gametes receive a particular type of gene. Either it will receive capital T or either it will receive a small t. The gene will not, sorry, gamete will not be going to have both capital T and small t. A gamete will be always going to have half number of chromosomes. Okay. So what does the law of segregation starts? So let me tell you here. So note down law of segregation. Note down law of segregation, please.
Done, sir.
Okay. What are the conclusions of dihybrid cross? Let's start studying the dihybrid. Okay, let's take the questions on this one. Just a minute. So here, if a question comes in your examination that how do Mendel experiment shows that traits are not intermixed. Okay. And there is a dominant trait and recessive trait. So how can you explain this one? So we can explain this one by saying that in Mendel's monohybrid cross, when F1 generation is obtained by cross pollination of homozygous tall plant and homozygous dwarf plant. Okay, when we are doing the cross pollination between the homozygous tall and homozygous dwarf, then during that time we were having all tall plants and there were no average plants. So this shows that the traits are not intermixed. Okay, they are not intermixed. Okay, they maintain their identity. Moreover, in F2 generation, we were having three talls and one dwarf plant. So one dwarf plant is indicating that we are going to have, means one dwarf plant is indicating that the traits or the genes are not intermixed. Okay, and they maintain their identity. Otherwise, if a small t has forgotten, if it is living with capital T, so it might have loosen its identity. Okay, but whenever it mix and it gives you small t, it means that they are maintaining their identity. Okay, they have not forgotten their identity of dwarfness. Got it? So here the question will comes in examination that how do Mendel experiment shows that traits maintain their identity or the genes maintain their factor maintain their identity and they are not intermixed. So we can explain it with the help of monohybrid cross. Okay, so note it down. Wait a minute. So Here is the answer for this question. Please note it down. Don't write it in bracket. This is statement, last statement. Just remove the bracket and add it here. Maintain their identity as dwarf plants appear in F2 generation. As dwarf plant appears in F2 generation.